no complication in analysis or understanding of such data. Then another way of data analysis and presentation for NGS data or high throughput data could be, for example, this is pathway plotting in different samples you can go for, or in different conditions. Then this is network level understanding. These are the hub genes, these are the hub genes, these are the hub genes or critical genes. In another way, you can say these are the genes which could act as a master regulator genes in any condition. Then another way of pathway network level analysis. So this is crosstalk, and this you could see how to analyze this pathway crosstalk in my paper, which was published in Cells in 2022 in this journal, which is like called GCIP software. And this is really very good software and much better than the software which is like these days commonly used from, and that is really paid software. So this is free software, and the other softwares which are in use, they're only better in the sense of easy handling and graphical presentation. But if you are slightly interested in a command line, then this software is really powerful software and highly advanced software. And this is real prediction of pathway crosstalk. Then if you are going to analyze the ChIP-seq data, the presentation of the ChIP-seq data could be like this. So these are, for example, enriched binding sites in 3 different conditions of the samples, and then enriched binding sites could look like this. So there are different ways of analysis. And if someone wants to go for chromosome wise, location wise, chromosomal location, and then the enrichment, transcription factor binding site enrichment, or the similar way of presentation will be applied for ATAC-seq data also, which is very common these days for clinical samples. So the another way of presentation, if somebody is looking for direct mapping of the changes on the genomes on the chromosome or on the chromosome, then this is really advanced way of annotated pictorial analysis, and this could be really one of the finest way of analysis. And there are really inbuilt tools or functions available in most of the coding platforms, which could easily plot the chromosomal mapping and these changes with proper annotation. There is no need to go for manual annotations. Then another way of presentation, if you want to be more interested in clarity, for example, for the purpose of your presentation in conference or paper, then you need to interpret your data with proper pictures, and this could be a really interesting way of presentation. And of course, it is time taken but easy to present. Then it is sample preparation. So for all the different NGS data generation, the sample preparation method could differ. So this is what you need to think. Then sequencing technologies remains the same, and the users decide which sequencing platform could be better in terms of finance, in terms of the quick delivery, and in terms of the quality of sequencing. But most of the sequencing platforms are highly competitive in terms of quality. Financially, they are varying. So this is what only matters. But quality remains very close to each other. Then quality control, you could check the quality of the sequenced samples by using FastQ software, and then use Trimomatic to remove the gaps. So another step is read alignment. Most of these steps remain common to all kinds of NGS data. Go for alignment, which I have explained in the previous lecture. Then if it is like, WGS, WES, then you need to go for variant calling. And this is a new step, which I have not explained in RNA-seq data. So this I will be explaining. So then again, annotation part, which remains the same for all the data, all NGS data in the end. Before you go for final analysis, you need to annotate the variations with the gene names. Then filtering, go for the quality, good quality data or predicted genes or mutations. So that is called here, you need to go for a statistical analysis. Then interpretation, which is very important. And here you need to read the research papers and review papers related to your NGS data type. And finally, visualization, of course, is very important. Like IGV viewer, UCSC genome browser, they are commonly used. Now, I'm coming to ChIP-seq data. So ChIP-seq data is basically chromatin immunoprecipitation sequencing, which is a powerful method to analyze protein DNA interactions. It combines chromatin immunoprecipitation with DNA sequencing to identify the binding sites of the DNA-associated proteins. And this is biologically very, very important. Then experimental design, as I mentioned, will change. And to prepare the samples for sequencing for ChIP-seq purpose, it is trivial. So here you need to little bit struggle in terms of sample preparation. Once you prepare the sample, then you proceed for the sequencing. So there are few steps. For example, this sample preparation includes cross-linking proteins to DNA, sharing the DNA into small fragments and preparing the chromatin for immunoprecipitation. And this immunoprecipitation is performed using antibodies to the protein of your interest. The antibody protein DNA complexes are then isolated by using magnetic beads or agarose beads. Then you send your samples for sequencing, which is like explained, which was explained earlier. Then again perform the quality check or quality assessment. The read alignment will remain the same in terms of, so basically for all NGS data, you need to assess the alignment, how many sequences where total sequences where reads were present, how many sequences were unpaired or not aligned, and how many samples were aligned. So all these things you need to align, and the maximum percentage of alignment means really good quality sample generated by the company or the platform. So which could, if you want to assess the alignment, then it could look like this. Now you could, if since we are in ChIP-seq data analysis, so pick calling is very important. And pick calling algorithms such as MAX are used to identify the regions of significant enrichment in the aligned reads, indicating open chromatin regions. So for example, if somebody is looking for transcription factor binding sites on the DNA, then they'll call it the enrichment region where this particular transcription factor is binding. So higher binding sites for a specific transcription factor means there is higher enrichment, and that is called a pick. So this pick could be aligned. For example, number of called picks here, this one. Pile of picks, this one. Tag after filtering in treatment, this one. then fit percentage, this one, and number of consensus picks here. So these are some of the things. I will show the how it looks like in the form of when you the plots. Then you go for motif analysis. So the signature, binding signature, how it could look like in terms of ATAC-seq. Then different motifs could have different sequence. So motif, sequence, or signature. And this could be analyzed by using common software. Now, again, annotation part is coming, like the analysis, everything will be done after annotation. So once you are finished with annotation, then you go for differential binding analysis, very similar to differentially expressed genes. Here, the difference between differentially expressed genes and differential binding analysis is that there you call it the number of genes, the genes expressed, the expression values. Here you call it how many binding sites are present. And then since you are comparing between different groups, more than one group, so more than one or two groups, so then you can compare, and that is called differential binding analysis. Similarly, the p